Hey all, welcome back to my channel. I'm Akanksha and today we are going to start with the second part of the topic diphtheria. In the previous video, we just learned about Corynebacteria, bacterium diphtheria, about all the features. In this one, we will be seeing about the lab diagnosis. Okay, first we will see specimen. We should remember there is a sequence of how to write the lab diagnosis. First, we will see what are the specimens required. Being a respiratory disorder and the throat infection, mainly it infects the throat and the pharynx, exudative pharyngitis and membranous tonsillitis. So first we will take throat swab. Okay, one or two swabs we will take, and that will contain the fibrinous exudates. Then second is a portion of the pseudomembrane formed, that we will take a portion out of it, and we know that there is the infection diphtheria also occurs in nose and larynx, and so we will take nose and skin specimen. If they are infected, then we will take the specimens from here. Then second, we will go for the direct microscopy. In this case, Corynebacterium diphtheria, we will be doing two types of direct microscopy. One is gram staining and the other is Albert staining. Albert staining is more specifically used for Corynebacterium diphtheria. So let us see what is written in, what will happen in gram staining and what will happen in Albert staining. So gram staining, it's a club shaped. Okay, what are the appearance we will see? It's club shaped gram positive bacilli, and we have already known that the arrangement is cuneiform or Chinese letter arrangement. Why does this occur? This is a snapping type of division, and this shape is coming due to the division of the daughter cells at an acute angle. Okay, so that's why this appearance is coming. They are irregularly stained. So this is overall how it will look in the gram staining. Next is Albert staining. Albert staining is more specific. We have just seen that Albert stain and Ponder stain, these are more specific or better stains or special stains which are used in this case for metachromatic granules. So Albert stain, this is more specific for Corynebacterium diphtheria. So what we will see, these are green bacilli with bluish black metachromatic granules at the poles. Okay, so this is the appearance, like green bacilli with bluish black metachromatic granules at the poles. So this is about the direct microscopy. Next we will go to the culture media. So Corynebacterium diphtheria, it is a fastidious, aerobic and facultative anaerobic organism. So this won't grow in the basal media. Okay, that will not grow in the basal media. So we need something different. Okay, so what we will do, we will incubate it at 37 degrees Celsius aerobically. Okay, in enriched media, what are the enriched media we are using in this case for Corynebacterium diphtheria? It's a blood agar, chocolate agar, Loeffler serum slope. Blood agar, what will be the appearance of blood agar? It will be small, circular, white, sometimes hemolytic, Mitis biotype. In Mitis biotype, we will see hemolytic. We will see later on the typing of the Corynebacterium diphtheria. In four types of biotype are present. Out of that, one is Mitis biotype. Okay, so in blood agar, it is small, circular, white, sometimes hemolytic, which is the Mitis biotype. Then chocolate agar, then Loeffler serum slope. In Loeffler serum slope, what will be the appearance? It is small, circular, glistening, white with yellow tinge in 6 to 8 hours. Okay, we will see the appearance in 6 to 8 hours. So what is the advantage? Advantage is that the growth detection can be done in 6 to 8 hours, and it is the best for metachromatic granules. And what is the disadvantage? Disadvantage is that this is a enriched media. Okay, so if we incubate it for more than 6 to 8 hours, then the growth of throat commensals will also occur, so we won't be able to differentiate it. So that's the different disadvantage. Now this was all about the enriched media. Now what about the selective media? Selective media is usually the best for the isolation of the normal flora. Okay, it's the best for isolation of the Corynebacterium diphtheria, because the normal flora will be inhibited. Okay, being a selective media, it will allow the selective growth of the Corynebacterium diphtheria. So what we use the potassium tellurite agar. Okay, potassium tellurite agar. So Corynebacterium diphtheria, what they do, they reduce the tellurite into metallic tellurite, okay, and this metallic tellurite will give the black color to the colonies. And this is also seen, we are seeing in the Corynebacterium diphtheria, this is also seen in Corynebacterium ulcerans, Corynebacterium pseudotuberculosis, okay. These two species also sees this type of black colonies, okay. And what we already read, diphtheria toxin, this these two species also has diphtheria toxin. Now what is the advantage of this potassium tellurite agar or the selective media? Being a selective media, it is a throat commensal inhibitor, so this is the advantage, which was a disadvantage in case of Loeffler serum slope. Now what is the disadvantage? Disadvantage is that the colony will appear after 48 hours of incubation. It's a long duration, so after 48 hours of duration we can see the colonies. Thus that's the disadvantage. So this is the potassium tellurite agar, and we can see the black colonies like this. Okay, so we just read about enriched and selective media. So what all we saw the specimen collected, throat swab and all, direct microscopy, gram staining and Albert staining, then the culture media in which we saw the enriched media and selective media. Under enriched media, blood agar, chocolate agar, Loeffler serum slope, and selective media, it is the one that is potassium tellurite agar. Next is identification. Identification, here we are doing three types of identification. One is biochemical test, and the other is automated, and the other is the diphtheria toxin. This is a special case for this type. So biochemical test, here it is His serum sugar fermentation test. Okay, biochemical test in this case is His serum sugar fermentation test, and automated identification is Malditov or Vitek. Malditov or Vitek, okay. Usually you can write it almost in all cases. Then diphtheria toxin demonstration. So diphtheria toxin demonstration can be done in vivo or it can be done in vitro test, okay, in vivo or in vitro. In vivo test, what we do is guinea pig inoculation, okay, guinea pig inoculation in subcutaneous and intracutaneous region. Then in vitro we have four types. One is LX gel precipitation test, detection of tox chain by PCR, detection of toxin by ELISA or ICT, hematochromatogenic test, then cytotoxicity on cell lines, okay. So these are the four types of in vitro test. So what is LX gel precipitation test? It's a type of immunodiffusion gel described by ELEC. Immunodiffusion gel which is described by ELEC, ELEK. Now what is done is the strain is isolated, then it is streaked onto the media containing a filter paper which is soaked in the antitoxin. Now the strain which we have streaked, okay, that is toxigenic, so that will liberate toxin. Now this toxin liberated will diffuse in the agar, okay. Now when it will diffuse in the agar, this will meet the antitoxin there in the agar, and that will produce the arrow shaped precipitation band. Okay, here see, if this is the filter paper, okay, and this is the agar plate. So three types of band we can see. One is crossover, okay, this is crossover. Crossover indicates unrelated strains, okay, they are not related. Then one is spur formation, means just touching, okay, this is the spur formation, that is partially related strain. And one is fused band, that means identical strain. So this is all about the LX gel precipitation test. What do you mean by LX gel precipitation test? It is a diphtheria toxin demonstration test or identification test for Corynebacteria diphtheria, and it's an in vitro test, and it's a type of immunodiffusion gel which is described by ELEC, and the procedure and the types of the three types of bands. Next, after this, we will see the typing of the Corynebacterium diphtheria. Now, typing is done to know the relativeness of the isolates. Whatever we have isolated, we will see how relative they are. Okay, we have seen four biotypes of Corynebacterium diphtheria. One is gravis, intermedius, mitis, belfanti. Mitis we have just read. In mitis biotype, we see hemolytic type in blood agar, okay, enriched media. The out of this four, the gravis is 100% toxigenic and most virulent, okay. And what does that help? These four biotypes, they differ in virulence as well as in toxin production. Okay, so that is how the typing is done. So here we have just seen the lab diagnosis, which consisted of the specimen, then collection, then direct microscopy, in which gram staining and Albert staining we have seen, then culture media, in which we saw the enriched media and selective media. Enriched media, blood agar, chocolate agar, Loeffler serum slope. Then selective media is potassium tellurite agar. Then we have seen the identification, in which biochemical, his serum sugar fermentation test. Then we saw about the automated Malditov and Vitek. Then we saw the diphtheria toxin demonstration, in which we saw in vivo and in vitro. In vivo is the guinea pig one, and the in vitro is LX gel precipitation test is the main thing, and the other three types. Then we saw the Corynebacterium diphtheria typing, in which we saw all the four biotypes. So this was all about the lab diagnosis. In the next video, we will see about the epidemiology and the prophylaxis, treatment,